Good afternoon, everyone, and welcome to the third webinar of the Anatomage Academy on the brain. This is Vanessa, and this afternoon I will show you how it is possible to use the Anatomage table in an effective way to explain this complex organ. The purpose of this webinar is to give you some examples from which you can draw inspiration for your classes. Therefore, before starting, I apologize in advance for any anatomical error. As I told you, I disable your microphones to avoid audio interference. If you have issues or questions during the webinar, you can use the chat, and I will answer at the end of the webinar. As you can see, I have already opened the high-resolution regional anatomy of the female brain. The brain is the central organ of the nervous system, and with the spinal cord makes up the central nervous system. The brain consists of 4 main regions. To better identify these regions, we can first apply a cut. So I select the clipping tool icon and I select the sagittal cut icon. And then I adjust the orientation to obtain this view. Now we can use the pen tool to highlight the different regions that compose the brain. The first one is the cerebrum, that is also the largest one. And now I'm using the green pen to highlight the border of this area. Then there is the cerebellum. I can change the color of my pen, and now with the red pen I highlight the cerebellum. The brainstem, that I can highlight in blue, for example. The brainstem is composed of midbrain, pons, and medulla oblongata. And finally, the diencephalon, that is this region in the middle. Now we can also, for example, use the arrow to draw an arrow and the text icon to add a text. So I tap with the finger on the point where I want to add the text, and then with the keyboard I can type the name of the structure. And then I can adjust the position of the annotation. You can do the same for all the regions, as I did, for example, in the preset number 3. This could be, for example, an assignment for your student. You can, for example, ask your student to identify the structures, color them, and then present the result as a screenshot. So they can take a screenshot using the camera icon. Now let me remove the drawings. So I open again the pen tool icon and I use the clear icon to clear the drawings. As we saw, the cerebrum is the larger section that composes the brain. As we can see, if we activate the histology, So let me first select the layout icon and select the histology icon. Here we will open on the right screen of our table another window. If I press now on a point on the cerebrum, I will open a list of histology scans available related to these regions. With this arrow at the bottom, I can scroll to see all the available histology scans. As we can see, for example, from this histology scan, the cerebrum consists of neurons and nerve fibers that transmit information. In this case, the neurons are highlighted by these arrows. In this other histology scan, we can also see the gray matter, here indicated with the letter C, that is the outer region of the cerebral cortex, consisting of cortical layers of neurons, and the white matter, that is this region, and is the deeper subcortical regions of myelinated axons. If we select the information icon, we can also see the description of the histology scan with also the annotations. On the other hand, if I press on a point on the cerebellum, I will open a list of histology scans related to this structure. For example, if I select this one and then I open the information icon, we can see that the cerebellar cortex contains 3 layers. The granular layer, indicated with the letter G, the Purkinje cell layer, highlighted with these 2 arrows, and the molecular layer, that is indicated with the letter M. Let me now go back to the previous preset, to the previous visualization that I have already saved. And from this visualization, we can see that the cerebrum is divided into cerebral hemispheres. The longitudinal fissure is the deep groove that separates the 2 cerebral hemispheres. Each hemisphere is conventionally divided into 4 lobes. Each lobe controls specific tasks. For example, we can select the frontal lobe. If I double-click on it, I can open the action menu, and through the action menu, I can apply a flat color to this region. For example, I would like to color the frontal lobe in yellow. And then, using this other icon, I can also turn on the annotation. Let's do the same also for the other lobes. The temporal lobe, as you can see, I just need to tap another structure to have the action menu related to that structure. In this case, I would like to color the temporal lobe in blue, and I activate the annotation as well. I do the same also for the parietal lobe, and finally for the occipital lobe. The lobes are named according to the skull bones that overlie and protect them. To activate the cranial bones, we can open the visibility menu. Let me first turn off the annotation using this a circle and remove the flat colors using the rainbow circle here at the bottom. And now I select the skeletal system and I select the checkbox next to cranial bones category to activate the cranial bones. Let me now close the visibility menu, and I would like to do the same as I did for the lobes. So I double click on the frontal bone, for example. I apply the yellow color, I activate the annotation, and in this case I would like to activate also the transparency using this icon to see the brain below the bone. And I do the same also for the other bone, temporal bone in blue. I activate the annotation and I apply the transparency. The parietal bone in pink. And finally, the occipital bone. Wow, now you can, for example, go back to the previous visualization that I have saved in a preset to make a comparison. Or you can also split the screen to have a dataset on the left screen and the other dataset on the right screen. To do this, simply press on the property icons, select the layout icon, and then select, in this case, the right arrow to move this instance of the table on the left screen, in this way. Now you can open again the table application. To save time, I have already opened the dataset with the cranial bones highlighted, and you can repeat the same procedure. So I select the property icon, I select the layout icon, and in this case I will click on the right arrow to move this dataset on the right screen of my table. In this way. Now you can interact with the two datasets independently. To go back to the full screen, simply press again the right arrow icon. Now I would like to focus our attention on the cardiovascular system of this region. Let me go back to the first preset. I open the visibility menu to activate the skeletal system, remove the nervous system, and inside the cardiovascular system, I would like to activate only the brain spinal cord arteries. Now I select the clipping tool icon to perform an axial cut using this icon. I would like to flip my cut, so I open again the clipping tool icon and I select this icon to cut the opposite side. And now I adjust the visualization, I adjust the orientation to obtain this visualization. Now we can, for example, double-click on the basilar artery to open the action menu. In this case, since the basilar artery is a vessel, we have the possibility to activate also the blood flow. If I press on the blood flow icon, we can see the blood drops flowing. If you select a second time, you can see also the vessels highlighted. And if you press another time, you will activate also the annotation of the vessels. As you can see, the carotid arteries supply oxygenated blood to the front and to the back of the brain. These two circulations join together in the circle of Willis. The table can be useful also to study in detail the cranial nerves. To this aim, I have already prepared a preset, the preset number 11 here, where I put the brain in transparency and where I apply flood color to some of the cranial nerves. The cranial nerves are the nerves that emerge directly from the brain. They are conventionally considered 12 pairs. In this preset, for example, I have highlighted in yellow the olfactory nerves that transmit smell information to the brain. I have also highlighted in red here the optic nerve that transmit information about the vision to the brain. In green, you can also find the oculomotor nerve that control the muscles that move the eyes. And finally, in light blue, the trigeminal nerves. That is a mixed nerve, but mainly with sensory functions. Thanks to the prosection dataset, with the table we can also visualize the same structure showed with the cadaver in a prosection dataset. So let me open the main menu, select the prosection icon, and in this case I would like to open the Brain 1 full dataset. In the prosections, you can find the annotation regions that are a surface color region with annotation and are available for all the prosections. Let me now adjust the brain in this way. And to activate the annotation regions, you can select these 3 bars icon. And now from this list, we can, for example, activate the right and left olfactory bulb. The left and right olfactory tract, the optic chiasm, and the left and right optic nerve. Regarding the cardiovascular system, we can turn on, for example, the right anterior inferior cerebellar artery that is located here in green. The right and left vertebral artery, the right posterior inferior cerebellar artery, here in light blue, and finally the basilar artery, that is here in yellow. These arteries are part of the circle of Willis. And finally, in this list, we can also find the longitudinal fissure that is highlighted here in purple and separates the brain into cerebral hemispheres. In the nerve pathway, inside the physiology section, you can also easily investigate the pathway of the nerves between the brain and the rest of the body. To save time, I have already opened this dataset. Let me now first move the first slider bar a little bit on the left to remove the skin and see the muscles. The gross movement, such as the movement of arms or of the legs, is generated in the motor cortex. Let me now zoom a little bit and select, for example, the right biceps muscle. In this case, as you can see, you can visualize the pathway of the motor nerve here in light blue. In this case, we have only the motor nerve pathway, as we can see in this dialogue. In this dialogue, you can also see the muscles, the nerves, the spinal cord tract, and the cortex region involved in the pathway. In this case, the cortex region is the left primary motor cortex that is highlighted here in purple. If you press on a point on the background, you can go back to the original visualization. Let me now activate again the skin, so I drag the first slider bar all the way to the right to have again the skin. From the skin, the brain will receive information about fine touch, pressure, pain, vibration, and temperature. Let's press on a point on the skin. In this case, from the dialogue, we can see that there are a different pathway. The sensory pain temperature pathway, for example, that is highlighted in red. The sensory touch pathway in yellow, or you can visualize both the sensory pathway, selecting this option. Again, as before, in this dialogue, you can see the nerves, the spinal cord tract, and the cortex region involved in the pathway. In this case, the cortex region is the right primary somatosensory cortex that is highlighted here in purple. From this visualization, it is clear that the cerebrum has a contralateral organization. The right side of the brain interacts with the left side of the body. While the left side of the brain interacts with the right side of the body. We can also study the brain using DICOM dataset. So let me open the main menu and select the case library. In the case library, if we select the segmentation tab and the brain icon, here in the middle, you can find a list of scans where the brain has been segmented. Let's open the first one. Let me adjust the orientation. This is an MRI scan. If I press the layout icon and then the 3D plus 2D icon, I can activate the 2D on the right screen of the table. In the 2D, you can adjust the brightness and contrast using this slider bar. And you can also move the plane of the cut using this other slider bar. As you can see, in the 3D, you have this orange plane that indicated where you are located with the 2D. If I select the visibility menu, in the third column, you can find the list of the segmented structure. In this case, you have only the brain, and if I press on the rainbow circle, I can activate some additional options. For example, the no clipping option. Let's flag this checkbox and then close the visibility menu. And now let's apply a sagittal cut. So I select the clipping tool icon and I select the sagittal cut icon. As you can see, I cut everything but the brain that has been segmented. And with the slider bar, you can scroll along the plane of the cut. In the library, also many pathological cases can be found. For example, if we select the specialty tab and then the oncology icon and the MRI filter, here in the middle you can find several cases of brain cancer. For example, this one is a case of a patient with a glioblastoma. Let's open it. Again, we can adjust the orientation and we can activate the 2D. In the 2D, we can use the slider bar to scroll along the plane of the cut, and here we can see the tumor. We can also apply a similar cut also in the 3D. So I select the clipping tool icon and I select the axial cut icon. And now I would like to apply another cut to have a slice in the 3D. So I select again the clipping tool icon, and in this case I use the freehand cut. So I draw a line along the direction that I would like to cut, and then I press on the side that I want to remove. And I move my volume to obtain a similar view. We can, for example, open the visibility menu to apply a different filter. For example, this filter allows me to better identify the tumor. And we can also adjust the brightness using this slider bar. Now we can, for example, take some measurements. So I select the measurement tool icon, the linear measurement icon, and I tap on 2 points on the 2D to obtain the measurement along this direction. As you can see, in the 3D, I have the same measurements. Let's open again the measurement icon to perform another linear measurement in another direction. Let me now remove this measurement, so I select the measurement icon and I select the topmost icon to remove the measurement. Now I would like to apply a different cut to see the tumor from another point of view. So I select the clipping tool icon, I restore the volume using this other icon, and I apply a sagittal cut in this direction. Now I would like to open again the visibility menu, and in this case I will select an ultra equality filters. For example, this one, the second one, that allows me to have a photorealistic visualization of the tumor.